Okay, welcome back to the Immunology Classics and Immunology Journal Club. And today we have a very special paper that just happens to be one of mine that, that I'm going to talk about today. But before we get started, don't forget to like the video and click on the bell and subscribe to the channel. And also scroll down and look at my media sites, social media sites. And go to my website and check on the writing tab particularly, and then you'll find all the papers, all the classic papers there. So you can have your own graduate seminar in immunology publications. So the paper I'm going to talk about today is, the authors were myself, MARGARET FAVATA, and Steve OROSZLAN. Title of the paper is The Production and Characterization of Monoclonal Antibodies to Human Interleukin-2. Strategy and Tactics. It was published in a Journal of Immunology in October of 1983, and we had submitted it in April of 83. The background on this whole paper is that I, as the principal investigator in our lab, in my lab, initiated this project and gathered the reagents, the cellular reagents that we needed to be able to do these experiments with. And I performed the initial experiments and to get my feet wet to figure out how to see if we could do it in our laboratory and so forth. And then I passed on most of the experimental aspects to Maggie FAVATA. Maggie was a really fantastic technician who had gone to the university. She was from the UK, gone to the university over there, and then emigrated to the United States and wound up in Hanover, New Hampshire. And I hired her on in my laboratory, maybe just a year or so before we started these experiments. Can't remember exactly. And she was just fantastic. And she was really good with the cell work that was required to make these fusions between the plasmacytoma cell lines and the mouse spleen cells in order to be able to generate monoclonal antibodies. Steve OROSZLAN was a protein sequencer person who was at the National Cancer Institute at the branch in Frederick, Maryland, and he was an aficionado of protein sequencing, which not everybody was able to do at that particular point in time. I had contacted several different people to see if we could get this done, because in addition to wishing to make monoclonal antibodies reactive with IL2, we were also trying to clone the cDNA encoding IL2. I knew that we needed to get the protein sequence. That was one way to get at cDNA. And so there were all kinds of things in the mix at this particular point in time. Now, the reason that we really wanted to make monoclonal antibodies and have them reactive to IL2 was multifaceted as well. But the driving force in this particular instance was we had gone through several years of biochemical separative experiments, taking crude conditioned medium from PHA or kinase stimulated lymphocytes that made the lymphocytes produce interleukin-2, T cell growth factor. But the problem was, is that we really didn't know what we were trying to do. But the thing was, we would make like 100 mLs of conditioned medium, which we thought was a lot, and then we would put it through separative procedures. And it turns out that each separative procedure that we put it through, we lost a fair amount of activity, so that we could end up. So we did a lot of analytical biochemical experiments on the molecule that was responsible for IL2 activity, and we could show that it was really a single protein molecule, had a molecular size of about 15,500 Daltons, and we knew its isoelectric point. And so we had really characterized the molecular basis for the biological activity. But we ended up, after we lost significant amounts with each fractionation, we ended up with pure IL2, but where we had minuscule amounts. And by this time, I figured that, well, the only way for us to do this was to get an antibody reactive to IL2 that we can make an affinity column out of, that we could then purify IL2. Now, this was just a year or two after we had developed the radio-labeled IL2 binding assay that identified the IL2 receptor, binding site on the IL2 receptor. And that helped us immensely because as I'll get into, it revealed to us that the amount of protein in a conditioned medium that we had readily detectable biological activity, the amount of protein was minuscule. So when we started out with only 100 mLs of this fluid, we basically didn't start with anything to speak of. And I'll get into that as we go into this whole process. So it turns out that, monoclonal antibodies had only been discovered or reported in 1975. So it wasn't too long ago. And for the most part, it took the world a while to catch up and to be able to do these sorts of things. It turns out that the monoclonal antibodies, and I reviewed that paper in our journal club. George Kohler was a postdoc with Cesar Milstein in London, and that's where they did the first experiments that showed that they could make these monoclonal antibodies. And George had done it to a very typical antigen. They made the antibody to the antigen, and the antigen was sheep red blood cells. Most people by that time, by 5, 6, 7 years later, they had repeated that experiment, and they could make antibodies to red blood cells and then to other kinds of cells and to lymphocytes and so forth. But there were few reports of people that had successfully made monoclonal antibodies reactive to individual proteins and also very small proteins. So George had been a graduate student in Freiburg University and had done his graduate work at the Basel Institute for Immunology in Basel, Switzerland. And of course, I had a really good friend there by the name of Max Schreier, who had been one of the first, if not the first person to make a helper T cell clone. And he was interested in how T cells helped the B cells make their antibodies, how they could become helpers. And he told me that making these monoclonal antibodies was an art form and that I didn't have a prayer in becoming successful to do this. But that didn't dissuade me, which was a very good thing, actually. So that's the background. And the intro, in the introduction to this paper, we go into the raison d'etre and the rationale for why we wanted to make these antibodies, in addition to using them to purify the molecules. We figured that polypeptide hormones or small protein hormones like insulin, for example. There was a fair amount of literature out there that said that if we could get an antibody reactive to our what I thought was a hormone, it would be very useful in our experiments to characterize the biological activity of IL2, because we could use the antibody in our in vitro experiments to neutralize IL2 activity and take it out, so that then we could do experiments with it and without it and so forth. And I mentioned the immunopurification. We also thought, we had the bioassay. That was our gold standard to be able to detect this biological activity. But that is in and of itself is not the end all, although it was a very sensitive biological assay, and we were really good at it by this time. We wanted to develop immunoassays, radioimmunoprecipitation assays, and ELISA assays, enzyme-linked immunoassays. These were the standard assays that most hormonologists, endocrinologists had used to develop assays for other kinds of hormones. We wanted to know, we thought that if we got an antibody that blocked or neutralized biological activity, that would help us identify the active site of IL2 and the site that was actually responsible for binding to the IL2 receptor. And finally, we wanted to really nail down the physiological role of IL2 in the immune response. The first order of business that we ran into is we needed a source of the IL2 in order to be able to get enough IL2, purify enough IL2 in order to be able to immunize mice, because that's what we were going to do. And of course, we were working with human IL2, and that was very, very good because of the fact that, well, first of all, most of our competitors were working in the mouse because they were card-carrying immunologists, and they didn't work in the human; they only worked in the mouse. But of course, because of the species differences in the amino acid sequence, so mice had IL2, and we knew that, and so forth. And we also knew from evolutionary biology that the ancestors of present-day mice and present-day humans had diverged 80 million years ago. So there had to be, so there was going to be differences in the genetic sequence that encoded the interleukin-2 protein, and therefore it would be a heck of a lot easier to generate an antibody in a mouse to human IL2 than if you tried to do it with mouse IL2. But of course, most of the card-carrying immunologists were trying to do mouse IL2 into mice to generate the monoclonal antibodies, because at that time, and it persisted for many years thereafter, the mice were the only ones that you could use, the only species you could use in order to make these monoclonal antibodies. So you took mouse plasmacytomas that would grow continuously, and that's what one of the things that you wanted to have. And you would fuse that with spleen cells or lymph node cells, but spleen cells that had B cells from immunized mice. And then basically then you would hope that the fused product that was then called a hybridoma would grow continuously, but also would make antibodies to which the immunized spleen cells were directed towards. So that was the system. And we needed a source of we had to, first things first, you have to immunize the animal. And so fortuitously, one of my former graduate students, Steve Gillis, had found a human T leukemia cell line that made huge quantities of IL2. And that cell line was the acronym for that cell line was JURKAT, JURKAT. And that was an acronym from the patient's initials. And it had been shown by this time that, and we knew, and we had repeated his experiments and shown that we could get much more IL2 from this cell line. So that was good. Then the next thing that needed to be done in this whole instance was to create immunoassays for the protein. And I'll get into that as we get into the results in this paper as to why we needed that and why that was important. That it turns out, just to give you the bottom line, that the affinity of IL2 for its receptor was so high that no antibody could really, that antibodies could not really compete very well. And so if the only assay you had to try to detect a hybridoma secreting a monoclonal antibody reacted with IL2, the only thing you had was the bioassay, neutralization of the bioassay. It was going to be, IL2 would win most of the time, and you might miss a lot of antibodies that would bind IL2, but would not necessarily neutralize biological activity. The other thing is that if you only had the bioassay to detect your hybridomas, the bioassay depends upon the proliferation of the target cells, the CTLLs. And if you put in supernatants from the hybridoma cultures that have been with hybridomas growing in them, most of the time, those growing hybridomas would deplete the media of all kinds of vital nutrients and so forth and so on. And so we would call that spent medium. So if you put spent medium on top of the CTLLs, what you'd find is that it would decrease the amount of proliferation of the CTLLs by comparison to your negative control. So that would give you a false positive. That was a problem. And the materials and methods in this paper, in terms of the cells that we needed, we had the CTLL cells, and we had a particular clone, 15H, that we used routinely for our bioassay. So that was good. We could grow them. We had the jerket cells, and we cloned this cell line, and we selected for high producers. And the clone that we used was just simply numbered 6.8. We needed to get the plasmacytoma, mouse plasmacytoma cells that would fuse with the spleen cells. And we got that from my good friend Ellis Reinhardt's in Boston. But that had been developed somewhere else. I think it was in Basel where they first used NS1. As NS1 turns out was a very good partner for fusing these cells to the mouse spleen cells. So those are the cells. The IL2 bioassay I've already gone through, described, we arbitrarily called 1 unit per ml of IL2 activity was in a preparation that you could have 50% of the maximum proliferation out of the cells at a dilution of 1 to 10. And it turns out after we got the IL2 binding assay that we could calculate out the molecular concentration, the molar concentration of IL2. We didn't have to use the units because units were just arbitrary. We made them up essentially. But a molar concentration has some chemistry involved in it. And we found that it was 10 to the minus 11th molar. That's really a high affinity. It's a low K equilibrium dissociations constant, but a very high affinity. So for the IL2 production and so forth, we used these jerket cells that we would harvest from exponential growth of the cells. We would then wash them and resuspend them in new growth medium at 4 times 10 to the 6 per 4 million cells per ml. We would then activate those cells with PHA and PMA. The combination was better than either alone overnight and in serum-free medium. That was really important. And then we would then take the cultures, spin out the cells, take the supernatant, concentrate the supernatant with an Amicon filter, we could then purify the IL2 from that supernatant with only 2 steps: gel filtration, molecular size, assaying for IL2 the whole time, and then isoelectric focusing, which would localize the IL2 molecules to their isoelectric point. And we knew by all of our analytical experiments that we then would get a single band on an SDS polyacrylamide gel, which was pretty close to being 100% pure. We couldn't say that yet because we needed to do other experiments to prove that this was actually a pure molecule. But that's what we used to generate enough IL2 to immunize the mice. So then, and the other thing we knew, because we were immunologists, that the immune response is dose dependent on the amount of antigen you use. And that's you've probably heard a lot about the doses of the vaccines that people are using for coronavirus now. And I think the standard dose of the Moderna vaccine was 50 micrograms or 100. And the one from the Pfizer BioNTech was only 30 micrograms. I got the Moderna, and I think that was a better vaccine than the Pfizer. So it's dose dependent. You can't just use a little bit. You have to know how much antigen to inject into the animals to get a detectable antibody concentration. And so that's sort of an empirical kind of thing. You know, we calculated out that we needed to give at least 10 micrograms of pure as we could get IL2 per mouse per injection. And we generated a lot of pure IL2 through this whole process. And we injected several mice at a time. So we had to generate a lot of IL2 to do that. And so the primary immunization we did with 15 micrograms. in the presence or mixed together with complete Freund's adjuvant, which is a mixture of oil and dead TB organisms together with your antigen that's suspended in an aqueous solution. And that's better than not using complete Freund's adjuvant to boost up an antibody production. So that was primary immunization. We would, after 10 weeks, we do a secondary immunization, just like in the coronavirus vaccine. Again, we'd use 15 micrograms per ml. And this time we would use incomplete Freund's adjuvant. And at the same time that we immunized them, or no, we'd wait 10 days, and then we would bleed the mice through their tail veins and to get a little bit of blood, and then test for antibody neutralization of IL2 in our bioassay. So we knew which mice of the ones that we'd immunized would have antibody number one and the highest concentration of the antibody, because we would titer everything and so forth. Then finally, when we identified one mouse that we were going to do this fusion with, we would immunize them a third time at week 12, 2 weeks after that week 10 immunization. This time we would give 30 micrograms suspended in normal saline. And then 4 days later, we would kill the mice, take the spleen cells, do this fusion, and then clone the hybridomas and first in microtiter plates and so forth, and then test after a pretty long period of time, more than a week or two, for antibody activity. Now, and so the tests that we generated in order to detect the antibody activity, number one was the neutralization, and we would test serial twofold dilutions of the serum from these animals, or supernatants from the hybridoma cultures. We also developed a radioimmunoassay using our radio-labeled interleukin-2 that we developed for the binding assay. We could, in tubes, we could incubate the radio-labeled IL2 with dilutions of the antibody. We could then precipitate the antibodies that were bound with staphylococcal A, that was on beads. And then we would spin those down and we would count the precipitate and the supernatant, looking for activity that would really come down with the staph A. And that was our radioimmunoassay. We also developed an ELISA assay whereby we would take our purified IL2 and just put it into microtiter plates on plastic and incubate for, for an hour at 37 degrees. And the purified IL2 would stick to the plastic through, I never quite understood it, but it was electrostatic forces. Or no, we do that for 18 hours at 37 degrees. And then we would then add bovine serum albumin for an hour to fill up any other sites onto the plastic that had not been occupied by IL2. And then we would add our antibody, and then we would add sheep anti-mouse immunoglobulin that was labeled with an enzyme that would create a visible color reagent. And then we could also use the spectrophotometer, but we could just look at the wells to find out which ones were positive if this would work. Then after that, once we identified a hybridoma, we had to then grow up huge amounts of these cells and allow them to make the antibody, so we could just harvest a lot of antibody. And we did that by injecting the hybridoma cells into the peritoneal cavity of animals. And we would take 20 mice at once, inject them with 5 million hybridoma cells, wait a couple of weeks, and they would blow up and have those malignant cells would grow in the peritoneal cavity, and those cells would make antibody the whole time we were there, that they were doing that. We would then, when the mice are still alive and so forth, we would inject needles, draw off the acidic fluid, precipitate the antibodies with ammonium sulfate, and then use them for our various assays that we were going to be doing to characterize these antibodies. Finally, after we would generate lots and lots of antibodies, purify the antibodies, we could connect those antibodies or bind those antibodies to solid phase absorbents. And there was different kinds that you could use. We used Afigel 10. We would mix Afigel 10 with 10 milligrams of monoclonal antibody, and they would undergo a chemical reaction coupled to the gel. Then we would use little TB columns with syringes with only 1 ml, and 1 ml actually is only at the bottom of the TB syringe. We would put 1 ml of the gel with the antibodies onto it, and then we could then drip down. Again, we go back to our supernatants that had the jerkit supernatants that had the IL2. We could allow the IL2 to attach to the monoclonal antibody column. We could then wash through everything else. Then we could just raise or lower the pH with 0.2 normal acetic acid, and that would then allow the IL2 to drop off, and we could do fractions and so forth, test them for bioactivity and test them for radioimmunoassay and also by ELISA. We could then take the IL2 that we got and we could get milligrams of the stuff then. I mean, this is so much better. A milligram, just to remind you, is 1,000-fold better than a microgram, and which is 1,000-fold better than an nanogram. And so we were finally in duck soup here with all these molecules of pure IL2, and we could then test them, take them to those next steps that we couldn't do when we were doing analytical biochemistry. We could now test them for how they behaved on reverse phase liquid chromatography. And we could also do amino acid composition and to test whether or not they were really only one protein, and also amino acid sequence analysis, which was the sine qua non at that stage. And one of the things we wanted to have to show that they were pure, but also one of the things that we wanted to use to see if we could identify the cDNA for this molecule. So those were all the techniques that we had to bring to bear. And the good news is Max was wrong. We were successful, and that's why we are talking about this paper right now. And he later told me that he didn't think I had a prayer in hell to be able to do what I did, and it didn't take me all that. It didn't take us all that long. You know, Maggie was great, and so there you go. In the results, and I think what I should do is go to the screen sharing right now. So in figure 1, this shows you a bioassay, an IL2 bioassay, where we're diluting out the sample of IL2 from 1 to 1 to 1 to 10,000. We have tritiated thymidine incorporation into the CTLL cells here on the y-axis. And this is the amount of IL2 we would get from normal lymphocytes, human lymphocytes, that we take from peripheral blood. And as I said before, it dilutes out, so it's about 50% of max and dilution of 1 to 10. But look at this. This is jerkit IL2. It dilutes out with half max at about a little more than 1 to 1,000. So there's a 100-fold difference in the amount of IL2 here versus here. And the quantitative aspect of this bioassay is one of the things that I've always said was instrumental in allowing us to go to the next steps. So when we immunize, so we we got the IL2 from Jerkett. We immunized the mice, and then we tested in the plasma of the immunized mice for anti-IL2 activity. So we would take the half maximum here, and that's the IL2 all the way across the top. And now we've titered into there increasing or decreasing concentrations of our plasma from the immunized mouse. And you can see that it, at a 1:5 dilution of the plasma, it was 100%, lost 100% of the biological activity, but then it diluted out pretty rapidly. So it's about 1 to 100 here, where it loses all of its antibody neutralization activity. Here, down here is the ELISA that we had worked out. And you can see this is control mouse plasma. And well, actually, that's control mouse plasma down here, and there's control mouse plasma up here, plus the IL2 in all the wells. And you can see with the ELISA, as we dilute out the plasma, we go out to a dilution of 1 to 5,000 before we finally lose our activity of the plasma to simply bind to IL2. So that was good. And then we went on, and in figure 4, so then we took the mice and did the fusion, cloned the hybridomas, tested the individual wells for IL2 activity, and we came up with 3 antibodies that we called DMS1, DMS2, and DMS3, and they're simply called for Dartmouth Medical School, 1, 2, and 3. And here is a neutralization of biological activity of DMS1 are the triangles here, and DMS2 are the squares, and DMS3 are the circles. And this is just control mouse immunoglobulin here. So you can see that we got neutralizing activity that was readily detectable at a reasonable concentration. Antibody concentration here at 10 micrograms per ml, but you see DMS3 does not have very much neutralization activity. We needed out 1 to probably almost 1,000 before we get half maximum neutralization. What we've done is looked at the ability of the hybridoma supernatants to block radiolabeled IL2 binding. And you can see, so here we've got biological activity in the solid circles, and we got binding activity in the open circles. And then this is control mouse immunoglobulin up here, so that you can see that we've blocked the biological activity at exactly the same concentrations that we've blocked the binding activity. And that gave us considerable delight, because then we figured that the reason that the neutralization of the biological activity was because it blocked IL2 binding. Then this is the effect of the DMS1 monoclonal antibody on the biological activity derived from the Jerkett cells, which is in the open circles, versus the rat splenocytes, which is here. So we have human and then rat. So it doesn't really effectively block rat IL2 activity. But normal human IL2 from normal human tonsils and mouse splenocytes, it works. So that was interesting. So it works against mouse IL2 and human IL2, but not against rat IL2. Now we look at the DMS1 monoclonal antibody. Inhibition of the CTLL proliferation is monitored by the tridethymidine incorporation. So this is where we have units per ml of IL2 and also then picomolar concentrations of IL2. And so the question is, if you use maximum concentrations of the antibody, can you overcome that by adding more and more IL2 into there? And you can see that works. Now, this is extremely important because this is the kind of an assay that other people, when they've tried to make IL2, and I'll get into that in the discussion, they didn't do that experiment. Then the other experiment that other people had not done when they were trying to do these kinds of things or to show specificity was to then take non-IL2 dependent cells and see whether or not their antibody preparations would block or would inhibit the proliferation of cells that didn't have anything to do with IL2. And so these are 3 different cell lines. One is HL60, another one is K562, and then mouse embryo fibroblasts, and the antibody activity going up to 500 micrograms per ml doesn't touch those cells, which shows that any neutralization in the CTLL assay was IL2 specific. Then in figure 9 here, we've looked at the amount of IL2, anti-IL2 activity of DMS1 in the open circles compared to DMS3 in the closed circles. And you can see, so now we're doing this as ELISA, just binding to the IL2 protein. You can see you dilute out DMS1 pretty fast, but DMS3 keeps on going for a long time, so that you got much better binding activity with using DMS3 versus DMS1, whereas DMS1 was a better neutralizer. So that was interesting. So we found that DMS3 was when we tried to make the affinity columns and purify IL2, and we tested them all, and DMS3 was the winner and would work very well. And in one step, we could purify milligram quantities of IL2 in one step through this column. So then we took that further, and we looked at it through HPLC, or reverse phase liquid chromatography. And this is the elution curve. Let me see if I can get my cursor. Lost my cursor. There it is. You can see there's a single peak of a protein coming off, as this is the gradient of acetonitrile. And you can see there's a single peak of protein, so that there aren't any other proteins. There's one protein in one fraction. That was good, because that's what we were hoping for. And then we could also then take the same preparations that we analyzed through that process, analyze them through SDSPAGE of affinity purified human IL2, 100 nanograms. So this is the IL2 right at 15,500 Daltons. These are the standard molecular weights. We also did 2-dimensional gels stained with silver stain, which is very sensitive. Should have picked up other proteins, and we always had just a single spot. Finally, we sent our best preps off to Steve OROSZLAN down at the NCI in Frederick, and he basically sequenced most of that protein by breaking it up into small peptide sequences. This is the first 15 amino acids through N-terminal amino acid sequencing, and that's what we needed to really prove that we had pure interleukin-2. So those are the data, and now we'll stop the screen share and go back. So all of these experiments were really a tour de force for us. We were really a small lab. There was me and Maggie, not too many other people, just a couple of us. So the discussion was, we figured that the key elements to produce these monoclonal antibodies, that this could be done with any interleukin that you wanted it to, but this time there were 10 or 12 more interleukins that were being characterized and so forth. First of all, you had to have a rapid and a very quantitative bioassay, and it's a very specific bioassay. You had to have cells that were producing as much IL2 or interleukin as you possibly could get. And you had to develop immunoassays to screen for these hybridomas, because the neutralization assay could lead to false positives. And you would also have you would miss an awful lot of stuff that was binding to your protein, but that you couldn't detect through that we're not neutralizing. So it was key in this instance for us to use this super producer clone Jurkat cells. And the other thing is that we could use serum-free medium. Cells are not too happy without serum, and you can't culture them for a long time, but all we really needed was an overnight culture, and so that worked well. So we could get enough pretty pure IL2 to immunize the mice with. We thought it was important to immunize with as much protein as we could possibly generate. Had to be in the microgram range. The immunoassays, the RIP assays, and the ELISA assay were crucial because of the fact that the affinity constant for IL2 binding to its receptor was difficult to neutralize with antibodies, because their affinity constants are anywhere from 100 to 1,000-fold lower affinity for binding to whatever they're binding to. So it was important for us to immunize with pure IL2 protein, because if we'd used a crude gamish, we would have gotten antibodies to other stuff. And then when we went to screen the hybridomas, we would come up with false positives. Now, there were two prior reports in the literature. So we published in 1983, was one that came out in 81 and another one that came out in 82. But the only thing they were able to show was neutralization of biological activity. They didn't show that competition experiment whereby you would titer in IL2 to see if you could block that neutralization. And they didn't use the specificity experiments on non-IL2 dependent cells to see if there was just non-specific inhibition and metabolism in their target cells. So they didn't really have it. And their interesting thing was one of the groups applied for a patent on their monoclonal antibodies, and they never came to fruition after that. We knew from our experiments and the fact that DMS3 was a very good binder, but not a good neutralizer, that it probably didn't react to the active site of the IL2 molecule. The immunoaffinity purification, where we could demonstrate beyond that what we had was really pure IL2, we could state that in whatever paper that we wrote subsequently from this series of experiments. And other people couldn't do that. We were years ahead of everybody else. That one of the reasons was consequence of this paper and this ability to purify large quantities of IL2 that then we could use then for further experiments. Subsequently, what happened was, the reason this paper was published in October of 1983 is that Tada Taniguchi published a paper that came out in March of 1983 with the cDNA clone. And of course, that cDNA clone, the sequence of the nucleotide sequence, then would predict the amino acid sequence. And the way he cloned the cDNA was using our mouse bioassay to detect the biological activity from his cDNA clone. And so that was a bummer, because we were also trying to do that. And there's a backstory to that I won't go into today. But it didn't make any difference in the long run, because we were so far ahead of, they still had to purify their stuff. They had to produce their recombinant interleukin-2 in large quantities. They had to purify it. They had to show that it was pure. Meanwhile, we just danced right on through. And some of the papers that I've already talked about, in particular, I think the most important paper was the Doreen Cantrell and Smith paper that came out in Science in 1984, where we used, and we could state, so we're using pure IL2 for these experiments. And this is what we found in terms of the biological activity and how the binding to the receptor and how the whole system works. And we found essentially that there were basically three fundamental things that had to happen in order to make T cells to undergo mitosis, DNA replication, and then mitosis. One was the IL2 concentration. The other one was the receptor density or the receptor concentration. And the third was the duration that you allowed the two molecules to interact. And we found using the flow cytometer that in a population of cells, all of which had IL2 receptors, there was 1,000-fold difference between the lowest, a cell with the lowest number of receptors versus one with the high. So there's a bell-shaped log normal curve. And so you look at that and you say, well, I wonder if it makes any difference if you have a lot of receptors or just a few. And of course it did. If you have a lot of receptors, you're faster. So you have to imagine that if you take a population of cells, it's like the New York Marathon. Everybody lines up on the Verrazano River Bridge or the Verrazano Bridge in New York City. And then the gun goes off and the whole crowd starts to go. But then they all spread out over time. And the real athletes are the ones that finish in 2 hours plus. And then 6 hours later, these stragglers come in. And so the ones with the highest receptors in this instance are the ones that get to the finish line first. And that meant and means that somehow on the inside of the cell, it has a way to count the number of receptor hits. And when it gets the right number, then it goes. And the other thing that happens is the cell then that starts to fire, does it all or none. It doesn't sort of replicate a little bit of its DNA and then say, well, you know, I don't want to do the rest of it today. And once it starts, it goes through the whole process to cell division and mitosis and creating two new cells. And that's an all or none process. And the word for that in cell biology is that it's quantal. And that whole process then led me to write several review articles and then finally a book called The Quantal Theory of Immunity. And it was all based on these experiments that we did back in the early 80s that really defined what was going on. So I will stop there. Thanks for watching. Don't forget to like the video, subscribe to the channel, click on the bell, check out the website, read the paper for yourself, and see whether or not that I'm, I'm just full of my oats or whatever. And thanks for stopping by.